Assalamualaikum and hi everyone, I'm Atira and now I'm gonna explain about evolution of fungi. Protists started to evolve since from 1.5 billion years ago. Plants, animals and fungi can all trace their ancestry to protists. And based on molecular data, animals and fungi share a more recent common ancestor than animals and plants. If you can see here in the simple phylogenetic tree of Eukaryota, we can see that the fungi and the animals share a common ancestor. So that's why fungi are more close to animals compared to fungi and plants. Fungi also are members of the supergroup of Opisthoconta alongside with animals, and common ancestor of animals and fungi was most likely an aquatic, flagellated, and single-cell protist. You can see also in the diagram of eukarya, protists, plants, fungi, and animals. We can see that the fungi and animals share the same common ancestors. So now, let us just focus on fungi. The classification for fungi phyla is still ongoing and incomplete. Currently, there are 6 phyla that have been recognized, which is Plastocladiomycota, Neocalimysticomycota, Tetridiomycota, Glomeromycota, Basidiomycota, and Ascomycota. And there's the 7th branch, which is representing the uncurtained lineage of the Zygomycota. But why Zygomycota is still in the uncurtained lineage? Zygomycota is not recognized under the current classification as the increasing evidence it was monophyletic, which is the Zygomycota may came from many ancestors. As you can see here of fungi phylogenetic tree, we can see Blastocladiomycota, Zygomycota, Neocalimysticomycota, Citridiomycota, Glomeromycota, Basidiomycota, and Ascomycota. We can see that in this diagram, the line for the Zygomycota is a dash line, as Zygomycota still have an arcane lineage. So that's all for me for the evolution of fungi. So for the next part, which is the structure of fungi, I will pass to my friend Banu.
I will continue structure of fungi. Okay, firstly, characteristic of fungi. Some fungi are single cell, such as yeast. Yeast is also found in soil, on plants, and even in humans. And some fungi are also multicellular. And these fungi are plant-looking like, such as mushroom and even molds. So this is the structure of mushroom. As you can see, this is the fruiting body. And inside of the mushroom, it is full of mycelium, and each mycelium has hyphae, which consists of cell wall. Okay, so this is some of the mushroom that I have found near my house area. So as you can see here, this is the cap, and when we zoom the cap, we can see the scales on the mushroom, and this structure is called ring. So when we look at this picture, we can see the gills of the mushroom in details. And this is called the stipe, or even you can call it as top. And this thing here, when we zoom it, it looks like this. And this is called the vulva. Okay, so the structure of fungi. The first is mycelium. Mycelium is also known as the body of fungi, which consists of a network of fungal. And this network is called hyphae. So as you can see in this picture here, the mushroom is full of mycelium. And you can see it has below ground mycelium, and also you can see the spores here. Okay, next is hyphae. Hyphae is a thin filament which makes up the fungus. It is long, thread-like chains of cells. Hyphae grow at the tips and the branch of the mycelium. It also gives mycelium large surface volume ratio, which maximizes the absorption of nutrients into body of fungus. So, as you can see in the picture here, this is mycelium. But when we look in details, the long thread-like chains of cell here is called hyphae. So next, septum. Septum are walls of tissue which divides the cell, and septum is also a porous cross wall. Septum, its function is partitioning hyphae into individual cell. So you can see in the picture here, it has septum here, septum here, septum here, which makes individual. Okay, so first, septate hyphae. So septate hyphae have pores, which allows cytoplasm and sometimes organelles to pass freely from one cell to another. As you can see in the picture here, this is the septum, which divides the cell into individual cells. And there is also aseptate hyphae. Aseptate hyphae have no septum, which makes no individual cell. So you can see in this picture here, there are many nucleus present in cytoplasm of a single hyphae. Next is cell wall. The cell wall of the fungus is made up of chitin. Chitin is a polymer of glucose organized into microfibrils, and each glucose molecule has a nitrogen containing amino acid group. So chitin is the same stuff. So this is the close-up of hyphae. As I said earlier, the septa is porous, which these holes allow some cytoplasm and even some organelles to pass freely from one cell to another. Okay, next I will continue with comparison between plant and fungi. So, both plant and fungi are eukaryotic, but plants are autotrophic, which carry out photosynthesis, while fungi are heterotrophic, which feeds on decayed matter. So, for motility, both plant and fungi are non-motile. As for organelles, each plant have one nucleus per cell, while fungi can have more than one nucleus per cell. So, plants' cell wall is made up of cellulose, while fungi cell wall is made up of chitin. And both plant and fungi roles in ecosystem is also different. Plants act as producers while fungi act as decomposers. And plants, its food storage form is starch, while fungi is glycogen.
Okay, so let's move on to the last part of evolution and characteristic of fungi, which is reproduction of fungi. Fungi can reproduce bisexually and asexually. Fungi reproduce asexually by fragmentation, budding, and spores. During fermentation, new colonies of fungi can grow from the fragmentation of hyphae. And mycelial fermentation occurs when fungi of mycelium separate into pieces, with each component growing into a separate mycelium. The second one is budding. Budding is a type of cytokinesis in mitosis. Somatic cells in yeast form buds. A bud forms on the side of the cell. The buds ultimately detach after the nucleus divides mitotically. The last one is the popular one is spores. Spores allow fungi to expand their distribution and colonize new environments. Spores are produced by one parent only through mitosis and genetically identical to the parent. Spores may be released either outside or within a special reproductive site called a sporangium. This is the life cycle of asexual reproduction during germination, mitosis, and this is the sexual reproduction that has three stages, which is plasmogamy, karyogamy, and gametagia. Sexual reproduction occurs when there is unfavorable conditions where fungi need to survive, so they undergo sexual reproduction. During sexual reproduction, hyphae from two different mating types make contacts and fuse, which is, during plasmogamy, haploid from two different mycelia fuse to form a heterokaryotic cell with two or more nuclei, which is a positive and negative type of haploid. This is what we call heterokaryotic stage or dikaryotic hyphae. The nuclei from two mating types, which is positive and negative type, fuse immediately and continue to divide until every cell has at least one of each nucleus. So a hyphae contains two paired haploid nuclei and N plus N called as dikaryotic. During karyogamy, the haploid nucleate fused to form a diploid zygote nucleus. Diploid zygote nucleus undergoes meiosis prior to spore formation when the nucleate eventually fuse. So meiosis takes place in the gametangia, which is the last part of sexual reproduction, in which gametes of different mating types are generated. Gametangia is an organ or cell in which gametes are produced that is found in many multicellular protists, algae, fungi, and gametophytes of plants. At this stage, spores are disseminated into the environment. So, thank you for sticking through with us.